I just I just put on the recording device. Sorry. Uh, Abigail is a graduate student at the University of Illinois, Champaign-Urbana. She is working with the team at the Illinois Natural History Survey on the project that connected Powderhorn Lake to Wolf Lake last year. After her presentation, we'll have a question and answer session. The presentation this evening is being recorded. It will be available within a week on Ollie's website. Would everyone please remain on mute and welcome Abigail.
Thank you, Michael. Like Michael said, I'm going to be discussing the Powderhorn Lake Wolf Lake Reconnection Project. And for our research, we've decided to call it From Isolation to Integration, Exploring Fish Movement and Stream Ecology in Recently Connected Lake Systems. Let's do a quick background into it. An important aspect of our study is urban ecology due to the lake's location. With the current population growth of the world, we are currently seeing about 50% of the world's population residing in urban areas. And within the next 40 years, it is estimated that 2/3 of the global population will reside in these expanding urban areas. And as these urban areas continue to expand, they will collide more and more with the natural environment that surrounds them. And as they do this, we'll see a more rapid and significant change of the ecosystems here. And this is where urban ecology comes in. Urban ecology refers to the interaction of organisms with each other and environments consisting of high-density residential and commercial buildings. The goal of urban ecology is to achieve a balance between human culture and the natural environment within cities. And this happens through the studying of biodiversity of the region, understanding how energy and material is exchanged throughout, and as analyzing all the ecological interactions within the cities. With this, there has been an increasing shift in perspective to begin extensive ecosystem restoration projects to stabilize, conserve, and balance ecological, climatic, and social sustainable development. Aquatic restoration in particular is important in urban ecology projects. However, there are many challenges that managers face when attempting to tackle these aquatic restoration projects, such as habitat degradation, altered hydrodynamics, loss of biodiversity, and pollution. Urbanization leads to habitat destruction, fragmentation, and alteration through the construction of roads, buildings, and other infrastructure, all of which can disrupt the natural water systems and disrupt natural flow patterns that ultimately affect sedimentation transportation, disrupts oxygen levels, and alters temperature levels throughout, ultimately reducing the quality of the habitat and leading to a reduction of biodiversity. Additionally, urban areas have the tendency to introduce pollutants such as chemicals, heavy metals, and nutrients into water bodies, whether through its direct dumping into the system or through urban runoff. These pollutants can harm aquatic organisms and degrade the water quality further. Now, there's a few methods to go about trying to combat these, and restoration and the creation of artificial habitats are a few. Often with the implementation of urban biodiversity planning, the implementation of habitat restoration, and the installation and maintenance of artificial habitats. In urban biodiversity planning, cities can play a crucial role in biodiversity conservation with the integration of things like green infrastructure, blue infrastructure, implementing more parks and green spaces, and installing more wildlife corridors, all to try to support urban biodiversity. With habitat restoration, there's many efforts to restore degraded aquatic habitats, and they usually typically involve removing some barriers like dams and just improving overall connectivity of the aquatic system. For aquatic artificial habitats, these can look like things like installing wetlands, artificial wetlands, installing and modifying fish ladders, modifying culverts, and installing and maintaining floating islands. All of these strategies can help a city contribute to their urban biodiversity, while also trying to attempt to tackle these aquatic habitat challenges. And like I said, a large part of aquatic restoration is restoring aquatic connectivity. Now, aquatic connectivity is the distribution and movement of water across the landscape, and it is essential for aquatic biodiversity and ecosystem processes. Many factors can affect aquatic connectivity, including water quality, land use practices, climate change, and invasive species. Poor water quality can negatively impact aquatic organisms and their ability to survive and thrive. And land use practices, including urbanization, but also agriculture and deforestation, uh, can shift the natural flow of water and disrupt the connectivity. Climate change can create alterations in precipitation, where we're seeing extreme flood events, and we're also seeing extreme drought events, all of which can, again, disrupt the connectivity of a system. Uh, Invasive species can come in and outcompete native species for resources and alter their habitat conditions through modifications. Now, when we talk about connectivity, we need to remember that there's two types of connectivity: structural connectivity and functional connectivity. Structural connectivity allows for the potential movement of aquatic organisms by physically connecting to systems, such as installing culverts. Functional connectivity describes the actual scale of dispersal between habitats, such as the number of fish actually passing road crossings through culverts. Now, with all of this in mind, let's shift to the study site. So quick background into our study site. Our study site is located in South Chicago, just north of Calumet City, and it is a very large and very urbanized region. Our study site features two lakes, Powderhorn Lake and Wolf Lake. Powderhorn Lake is a 50-acre freshwater lake with an adjacent 55-acre shallot pool and is managed by the Forest Preserves of Cook County. Wolf Lake is a 950-acre freshwater lake that straddles the Illinois-Indiana border right along this highway and is managed by the Illinois Department of Natural Resources and the Indiana Department of Natural Resources. Now, Powderhorn Lake is one of the few remaining examples of a dune and swell topography that used to characterize the Calumet region along the south shore of Lake Michigan. These two lakes used to be part of a large collection of marshes, lakes, and rivers adjacent to Lake Michigan, but over time became more isolated by urban and industrial development and were exposed to years of environmental damages. Now, the motivation of this project was there's a big issue with such a big lake being located in a city, and that is flooding. During high rain periods, Powderhorn Lake would overflow into nearby residential areas, causing public and private property damage. And this was a problem for the city. They couldn't have that happen every time there was heavy rain. So they proposed connecting Powderhorn Lake to Wolf Lake to safely funnel the overflow of Powderhorn Lake into Wolf Lake to protect these residential areas. Now, this came with additional possible benefits of enhancing fish and wildlife habitat, possibly improving the hydrological connectivity of the region, and possibly reestablishing and restoring more than 100 acres of emergent and wetland habitat. Though the project was proposed in 2020, due to COVID, construction was delayed until 2022. And that construction involved installing a series of culverts, a stream channel, and a small pool. So here you can see Powderhorn Lake. What you can't see is underground. There's a bunch of culverts that connect it all the way up to this stream channel that they dug. And underground, there's more culverts that connect the stream channel to this little pool that they dug in the parking lot of Wolf Lake. And that parking lot is right about here where it connects to Wolf Lake at the boat dock there. And on October 23rd, 2023, the connection between Powderhorn Lake and Wolf Lake was completed, and water from Powderhorn Lake was allowed to flow into the intermediate habitat, so into the stream, and then into Wolf Lake. Water levels of Powderhorn Lake and the intermediate habitat are being controlled by these water control structures as depicted. This is in the marshy region north of Powderhorn Lake. And as water levels rise, they rise and rise and rise until they meet this threshold, and then they fall through this water control structure, which reduces the level of water within the lake, which helps solve their flooding issue. Now, as I said, a large component of the connection between the lakes is culverts. However, studies have found that more than 2/3 of culvert crossings are not fully passable to aquatic organisms. The size and shape of a culvert can affect the direction an organism travels, as some facilitate upstream or downstream movement depending on the dimension of the culvert, such as lengths, angles, and slopes. Fish, like people, have different strengths and weaknesses, with some of them being able to swim in harsher conditions than others. However, no matter what type of fish it is, all the fish will face several environmental challenges, such as low flow, low depth, variable temperatures, and poor water quality. Additionally, there's a chance of vegetation and debris buildup that might block the entrance of the culvert and prevent passage. So now that we have structural connection established and the residential flooding is no longer a problem, we have a unique opportunity. There is very little research on reconnected lake systems, and today I will be discussing some questions we hope to answer within this system. First, we want to know about the fish movement. Which fish are moving? Where are they going? How far are they going? And what's driving their movement? So there was a wide variety of fish in the system. There's minnows, there's crappie, sunfish, bass, all different types of fish. On the slide, I've shown some of the fish that we've collected while we've been out and sampling. So here you have a lake chub sucker, golden shiner, flat-nosed minnow, even some trout out there, an Iowa darter, some killifish, and some assortment uh, of sunfish and yellow perch and whatnot. So there's a lot of different fish species out there. With this connection, the fish have a couple of choices on which direction they move. They can attempt to go they can attempt to move upstream, which would mean they are moving from Wolf Lake to the pond, to the stream, to the marshy region, and then to Powderhorn Lake. Or they can go with the flow and move from Powderhorn Lake to the marsh, to the stream, to the pond, and then to Wolf Lake. Now, there's a couple of things that can determine the direction and distance a fish decides to go. And for non-random dispersal, it's been shown that disturbance or body size has been heavily or highly influential on this distance and direction. A disturbance, like an extreme flow event, can increase overall downstream dispersal of smaller bodied fish. So in this scenario, small fish are more likely to be forced to go from Powderhorn Lake into the stream passage. Though smaller bodied fish and juvenile fish have been shown to make attempts to move upstream in other systems, the probability of successful passage is reduced when you compare to their adult counterparts who have also been found to travel longer distances in those systems. Regardless, all fish face several challenges, as we've talked about, like water velocity and depth, and this kind of can influence how the fish travels through the passageway, as some fish may not be able to swim against strong currents or through deep water. Now, we are out collecting data at each site monthly from May to December. And for the fish specifically, we are sampling in May, July, and October and December to see kind of seasonally what's there. And the fish surveys are being conducted using a DC boat electrofisher. You can't see the boat, but that's what these two little anodes are attached to. And bike nets, which is kind of what this picture is showing, and the Smith Route 24 LR24 backpack electrofisher for the stream. Electrofishing is a technique used to sample fish populations by sending an electrical current that temporarily stuns a fish, making them easier to capture. The technique, when applied correctly, doesn't harm the fish, but only stuns them temporarily. And in this, you can't see the back end of the boat, but these two anodes are connected to a generator that sends out an impulse through these wires into the water, and the fish get stunned and they float to the top of the water where they get scooped up and are put in the boat until they can be measured and ID'd. For the spike nets, the fish can swim. If they swim into the net, they keep going forward and through the hoops when they go through the hoops, they can't get back out. With the backpack electrofisher, it has a very small version of this large pole that the user kind of just waves back and forth in the water as fish pop up and get scooped. So all these fish are being collected, they're being identified to species, and they're being measured to total lengths in millimeters, and then weight in grams. I put that on there twice on accident. My bad. Uh, all the fish are going to be scanned for the presence of a passive integrated transponder tag, and these PIT tags tell us if the fish has already been sampled before because we scan it, and if it's in our data, then we can see that we have it. And if it doesn't have a tag and it's greater than 75 millimeters in length and appears to be in good condition, we will implant it with a tag. Again, when done correctly, this doesn't harm the fish. Uh, and we're implementing them with these PIT tags because we have placed 4 radio frequency identification LITS cord antenna systems at each of the connection points. So this is kind of what it looks like. So here's the actual antenna cord. It goes into the water in front of the connection point so that when fish swim from the connection, they will have to cross over it. If that fish has a tag, the system is scanning for these tags at a rate of about 10 scans per second. Whoop, I went the wrong way, my bad. And this cord picks up that, and it's being powered. You can't see it, but there's a box back here that kind of has like the brain of the computer, and it records the data and it stores it until we go out there and we collect it. It's being powered through solar panels with backup power with batteries. And we have 4 of them out there. We have 1 at the marshy region between Powderhorn and the marsh. And then we have 2 within the antenna, within the stream at each connection point. And we have 1 at the boat ramp, Wolf Lake. This is so we can capture all the movement between, because if a fish goes from Wolf Lake into this little pool, but doesn't make it to the stream, well, this antenna tells us that happened. If a fish goes from Wolf Lake into the stream, but not into the marsh, we can have it recorded here. Or if a fish makes it to the marsh, but not to Powderhorn, we will have that data. So we're pairing this fish data with environmental data, which is why we're going out monthly from May to September. And in this, the first thing we're going to be doing when we go out there is we're going to document and record any vegetation or debris buildup at the connection points, and then we're going to remove that buildup. Because again, we're trying to facilitate or allow the movement of organisms between the system, so we don't want anything to bar the movement. And then we're going to go out and we're measuring water clarity, temperature, dissolved oxygen, phosphorus, chlorophyll A, water depth, and flow. For water clarity, we have this special disc that it's called a Secchi disc that you lower into the water at a set rate. When you can't see it, you pull it up a little bit, and that can tell you how clear the water is. For temperature and dissolved oxygen, we're employing a couple of methods to get this data. We have these YSI field optical probes where you lower at a set rate again, and then it gives you a gradient of the dissolved oxygen and temperature throughout the levels of the systems. For total phosphorus and chlorophyll, we are taking water samples from a single fixed index point where we lower it all the way down, and then we collect it up, and that gives us a kind of a profile. And for the water depth and flow, there's engineers out there who've installed these special flow meters, and that will kind of tell us this information on a continuous basis. And we've been doing this for a little bit. Between this last fall, we went out and we tagged some fish. We have a total of in 2023, we had a total of 490 fish tagged, 322 in Powderhorn, 38 in the stream, and 135 in Wolf Lake. We set out 1 antenna at the stream habitat in September of 2023 because we were just trying to test the equipment, make sure everything was functioning the way we expected it to. And we left it out there for about 4 months and pulled it in early December. And over that period, we detected 17 individual fish. In September, we had 2 unique detections with a total number of 202 detections. All this is saying is that we had 2 fish that swam over that single antenna or swam near it at enough where that the antenna picked those 2 tags up 202 times. In October, we had 18 detections when unique detections. But if you remember, I said we only had 17 individual fish identified. This means that there was one tag that we picked up that didn't belong to our data set. And we don't find that all that surprising. There's a lot of projects out on Wolf Lake and Powderhorn Lake, so we just picked up somebody else's tag. And we only had 109 detections, so they didn't really stick around the antenna all that much. In November, we had 10 unique detections with 5,814 total detections. That looks amazing, except about 5,200 of these were a single black bullhead just sitting on the antenna for quite a long time. In December, we only had one detection, and we only detected that fish 5 times. But again, we did pull the antenna pretty early in December, so that might be part of it. Just last month, we went back out and we tagged some more fish. So we've increased our Powderhorn sample to 389 fish, Wolf Lake sample to 241 fish, and our stream sample 273 fish. So now we have a total of 803 individual fish tagged across 25 species. And again, we have a range of fish based on each species, but basically, if it's a good condition fish and it's greater than 75 millimeters, except for minnows, because minnows don't tag that well with these fish, with these tags. We're basically tagging everything and anything we can get just to see what species are going to try to use this connection. Here's a quick layout of when we're doing things. We download the fish movement data, so the antenna data monthly, and we are going out over 4 months for the electrofishing and only doing a fall for the pike net. We're doing the water quality data monthly. So we're also going to be looking at this new habitat because this entire plot here used to just be shrubs and trees and grass. This water body didn't exist. They went in and dug out this entire channel and just made this stream. So with this, we want to know what aquatic animals will colonize this new stream channel. We also want to know will only environmentally tolerant organisms use this channel. So for this, we're going to be using not just fish, we're going to be using benthic invertebrates, aquatic insects, and zooplankton. And we got to keep the fish in there. So we're going to be using all those. So as a part of the connection, like I said, that new stream channel was dug, which means it's a novel ecosystem because it's a severely changed with its abiotic conditions and its biotic composition. And because of its location, it can be classified as an urban stream. And a feature of urban streams is they typically have limited diversity and they typically show frequently disturbed communities. Now, in the regular development of a stream, how it goes is you have established a substrate diversity with a higher diversity of substrate, usually leading to a higher diversity of organisms in it. And in this, over time, things keep moving in and out, but over time, it's expected that the community variability will decrease, with early colonizing species not typically persisting. But again, this is an urban stream, so that's not exactly what's going to happen here. With the regular possible disturbance events, like the regular periods of flooding or higher volumes of water, we are expecting to see a more stochastic colonization dynamic with it's just not going to be as stable as you'd see in a typical stream. So to do this, it's going to be very similar to what we're doing with our fish sample. We're going out monthly, May to September. We're going to be collecting zooplankton, benthic invertebrates, aquatic insects, adult fish, and larval fish. For zooplanktons and rotifers, we'll be using this Wisconsin 63 micron zooplankton net. And how you do that is you dip it into the water, trying to keep it straight, and then you pull it up to try to get a complete column of the water to try to get the zooplankton that are in that column of the water. For the benthic invertebrates and aquatic insects, we're going to be using a D net. We're going to be using the kick net method. And basically, you put the net down on the substrate and you kind of kick the substrate in between and you swish the net back and forth, trying to collect anything that's in the substrate or in that area. for larval fish, so these are the little baby fish, we're going to be using a modified larval fish net push where we're just dragging this net that kind of looks like this across the body of water to try to collect any young fish so we can see kind of what's in there. And like I said, the adult fish are going to be surveyed with the Smith Route 24 electric backpack shocker. Excuse me. The larval fish and the adult fish transects are going to be one complete pass of the stream to just try to gather everything up in there. The fish are going to be the adult fish are going to be identified in the field, and again, they're being measured to millimeters, weights, and we will scan for the pit tags, but basically we just want to know what fish are there. The zooplankton, rotifers, benthic invertebrates, and larval fish are going to be enumerated and measured in a laboratory setting, and we hope to ID them down to genus, but some might be a little tricky. We have high hopes with that. We're also going to be using an IBI indicator, which just kind of tells you the overall health or the water quality or the quality of the system. And for fish, it looks at things like richness, evenness, diversity, abundance, percent tolerance, percent intolerance, and a few other things. And it compares it to the water quality or environmental factors, and it kind of matches up to see if there's high diversity. Usually indicates that it's the condition of the stream is good, and if it's low, it tells us that it's a harder system to live in. So very similar to our sampling protocol for the environmental conditions for the fish movement question, we're going out monthly, May to September. We're going to start by recording and removing the vegetation and debris. We're going to measure the water quality, temperature, dissolved oxygen in the same manner with the Secchi disc and the YSI probe. And then we're going to collect our water samples to get the concentration of total phosphorus and chlorophyll A. But in this, we're going to visit we're going to do it at one site within it, just try to get an idea of what's going on in the stream. Here's kind of when we plan to do everything for this. Basically, we're going to collect macroinvertebrates and zooplankton monthly and our water quality data. We only are collecting larval fish pretty early on because after a set period, they just don't spawn as much, so they're not expected to be seen. And we're going to follow the same type of timeline for the sampling of the adult fish. Here's a quick visualization of the sampling map compared to like the actual site. For each site, here's Powderhorn Lake. The index site would be just about here. And at this index site, this is where we're collecting our temperature and dissolved oxygen profiles. And this is where we'll collect our water sample to just pull it up and get it there. Then we have our electrical DC electrofishing pushes. We have 3 of them. We are kind of staying away from this part of the lake because the depth there is just not very nice. It's hard to get anything out there, so we're just kind of sticking to the southern portion of the lake. And here's the marshy region. We have another connection point, and there's a flow meter and a hobo logger at each. And just, yep, it's a lot. We're getting a lot of data from a lot of different spots throughout the systems. And I guess with that, before I finish up, I'd like to acknowledge our project funding from the Illinois Department of Natural Resources and the Forest Preserves of Cook County. We'd also like to thank Steve Sillick and the Forest Preserves of Cook County for assistance with project coordination and equipment, and then David Ife for all of his help with field work, and he's been very instrumental throughout it. And if anybody has questions later on, I can be contacted at that email. And yeah, with that, I can answer questions if people have any.
Hello, how are you doing?
Good, and you?
Okay, I just want to ask you if you all deal with rare fish such as the Iowa darter, banded killifish,
Yep.
and lake sturgeon.
Yeah, we haven't seen any lake sturgeon, but we have come across a single Iowa darter this last time we went out. The killifish, we get them a little bit more when we are out sampling. We do mark and record every instance of every fish we get. So when we release all the data and all that, people will see all the fish we've sampled throughout.
And what about for the starhead, for the starhead top minnow? Have you heard of that type of fish before?
I've heard of it, but we haven't seen any in the system, and I don't believe it's been recorded in the system before.
Any bowfin and other rare fish in the Calumet area?
I'm not sure about the Calumet area in itself. We haven't seen any in either the lakes since we've been working on it. So just yeah.
So the size limit on the pit tags is 75 millimeters?
Yes.
Okay. Where were the pit tags, were they inserted? Were they inserted in both Powderhorn and in Wolf Lake, or only?
We're implementing them in Powderhorn, the stream, and Wolf Lake, basically to see what moves and like which direction they're going from. Because when we went out there, was already fish in the stream, so we're wanting to see if they're like sticking around the stream or if they're leaving the stream, and just having them ping out will tell us.
It would seem that an effort to get a more equal number of pit tags per species would be useful. There's some species that are way overrepresented, and then some species with a single tag.
Yes, we did run into that problem. Our main goal is to, when we go out there, we try to get as much of everything that we can possibly get, but with sampling, we're not always that lucky. We just get what decides to pop up, but we do put in a decent amount of effort to try to sample as much as we can.
Okay, doesn't the effort of removing the vegetation indicate you'll have a somewhat artificial result? That is, when your study's over, nobody's going to be removing the vegetation.
We aren't sure if the vegetation is going to build up, but if it does build up, we are going to document that and record it so that future managers know that this issue might pop up for this type of management strategy, and it is something that would need to be considered for future incidents.
And just to follow up on that, Abigail, I appreciate this great study here. The vegetation you're removing is kind of things that have maybe floated up and then blocked the culverts. You're not like removing, rooted vegetation elsewhere in the lake.
Yes,
it's just
that's correct.
Clogging up the system.
Yeah, we're just trying to make sure that nothing clogs it up and just that water can keep flowing. We don't expect it to be an issue, but with everything, fieldwork can be all over the place. So we're ready for it if it is.
So this Progress 2024 slide you pulled up again, when you say this 2024 has just started. So is this 2023 data actually?
This includes compounds with the 2023 data. We went out just a couple weeks ago and sampled the fish communities and implemented more.
So this is through May, the May data in 2024. Yes. Then you'll continue monitoring and sampling through this year. Will there be a 2025 collection, or is it just through 2024? Yes. It will be 2024.
We plan to do. Yes. Great. I pulled this up because of the questions on what species, just so people could see it and have it easier to see.
And then 2025 will be your final data collection year, and then you'll be writing your scientific articles and whatnot.
Oh, yeah. Hopefully we'll get it out quickly for people to have it.
Great.
Abigail.
Yes.
We have cameras out there to how many and what other functions?
We personally don't have cameras out there. For our project, we have just the antenna systems set up.
Okay.
So I'm unaware of the cameras, but if somebody else is using them, they can just kind of see with the like if there's cameras set up on the stream, you can see like what types of birds or what types of mammals or other wildlife decides to use that habitat. That'd be really cool. That's just not something we're doing.
Okay.
Are you monitoring the water levels in Powderhorn and in Wolf Lake?
Yes. Or just seeing the depth over time. We're going to compare
The
that.
water level in Powderhorn seems extremely high since the system was installed.
Yeah,
I've been
there's
over there a number of times, and it seems like it's raised the level in Powderhorn rather than I expected it to decrease it.
That's because the water level has to raise to a set point before it will the system will flush out. So it's allowed to raise and lower artificially on its own. They have the control structure closed at the moment, but whenever they facilitate the water moving, it's a man-made facilitation.
Okay, other questions?
Yes, I have a question. It has to do with water level. Some of those slides looked as if there was not enough water flowing in the streams between the other bodies of water. So that would mean that at certain times of the year, the fish can't move. Is that correct?
That is correct. With the limited flow, that's why we're going out more on a seasonal basis to see over the periods of time what occupies that. And that's why we have an antenna at the marsh and at the streams, like between Powderhorn and the stream to see when fish are being able to move. So we'll be able to document that as well.
Thank you.
Are there other questions?
On connectivity, are you worried about the Asian carp?
There right now there's only
I mean, that's Corps of Engineers is trying to separate the Illinois River from Lake Michigan, and you're talking about increasing connectivity.
Well, with this, we've only connected directly Wolf Lake to Powderhorn Lake. We haven't connected the connection isn't between the other lakes, but in the system we've only identified common carp in it, which they're in the system. We hope they don't like go other places, but with the flooding that was already occurring, there was fish were already moving a little bit between. So we're just trying to, this whole thing is controlling the water level from flooding into the residential areas. So it's just, bloop.
I have another question. It has to do with the beaver. I mean, there are beaver at Wolf Lake.
Yes.
They come up north to Eggers and its marshes. So well, your study obviously isn't tracking beaver activity, but do you need to monitor where they are for your?
We did have to modify our antenna system because when they were out, the antenna was out in that 4-month period. It was damaged by, we believe it was beavers. There was just big chunks taking out of it. So we've gone through and insulated the cords, well, not insulated, but we wrapped them basically with this heavy duty plastic. And then we tested to make sure that it was still able to read the tags that would swim over. So we're hoping that this prevents them from trying to eat our antennas anymore, because that was not, that's not great.
Thank you. Thank you.
Yeah.
Abigail, I just want to tell you that this was a very informative and very lucid presentation. I am not a scientist at all, but you made it very, very accessible. So to people like me. So thank you.
Other questions? If not, I just want to express my gratitude to Abigail for her presentation this evening. Valerie
Thank
and
you
I,
for having me.
okay, Valerie and I thank you for attending this evening's gathering. As in past years, we will be taking the summer off, and we'll see you on the first Tuesday in September. So thank you everybody for coming out, and we'll see you in September.
Have a great summer, everybody.
All right. Thank you.
Adios.
Okay. I can't see the recording. Where is the recording? Can't see it. Can't see the recording. What's this recording?